Hello friends, welcome back to our YouTube channel Doctor's Corner. In this today's lecture, we are going to discuss one of the very important topic in the neuromuscular physiology, that is neuromuscular transmission. That is how the impulse from the neuron or the nerve is transmitted to the muscle, okay, more specifically in a skeletal muscle. So it is we are going to discuss this under this heading neuromuscular transmission. And from exam point of view, in the subject physiology, that is in the first year MBBS, you will get a 10 marks question regarding the events which occurs at the neuromuscular junction, or what are the events in the neuromuscular transmission. Also clinically, it is very important for the various diseases like myastheniagravis, Lambert-Eaton syndrome. We'll discuss about that disease and other drugs which are acting on the neuromuscular junction. But before that, today mainly we are concentrating on this topic, neuromuscular transmission. So this, what do you mean by neuromuscular transmission is, suppose this is a biceps muscles here, right? Suppose if I want to contract my biceps muscle, the message from my brain ultimately goes with the help of the neurons to the spinal cord, and from the spinal cord, that is the anterior horn cells of the spinal cord, which is a motor neuron, it is the alpha motor neuron, will transmit this muscle to the concerned muscle. Okay, now what are the events that takes place at this neuromuscular junction? How this signal, so in the nerve, the signal is transmitted by the form of action potential, which we have already discussed in the last class. That is an electrical phenomena through which the signal passes in the form of action potential, and this action potential is converted into chemical energy. That is it will convert that into release of some chemicals known as acetylcholine, which is a neurotransmitter, and that acetylcholine causes the contraction of the skeletal muscle by causing depolarization in the skeletal muscle. Okay, so how exactly this electrical event is converted into chemical event, and this chemical event is converted into one more electrical event, that is the signal from the nerve goes to the muscle. Okay, so there are so many events, so please give your kind concentration how exactly it will transmit the electrical, it is the action potential is converted to the action potential of the muscle. So before that, we'll have a idea about the structure of the neuromuscular junction, how it looks. So where the neuron or the neuronal terminal end, okay, this is the neuron which will be ending at the muscle, that is this junction, it is called as where the neuron that is the axon, that is the terminal end of the neuron ends and unites with the muscle, it is known as neuromuscular junction. So what is the structure here? So this space between this, it is called a synaptic cleft. Okay, this pinkish color thing what you are seeing is a muscle, this yellowish color thing is the axon or the nerve, and this is a myelin sheath. So this space, it is called as neuromuscular junction, and this space, it is called a synaptic cleft, which is around 20 to 30 nanometer. Now in this neuronal endings, you can see there are mitochondria, and most important thing, this vesicle like structure known as synaptic vesicles, which contains a very important neurotransmitter or chemical which is called as acetylcholine. Okay, how this acetylcholine is released into this synaptic cleft, and this causes or send the signal to this muscle receptor. There are muscle receptors which are present, which are known as nicotinic acetylcholine receptors. And what are the enzymes which will act on this acetylcholine? We are going to discuss in this today's video. So I'll draw a line diagram to make this point more clear. Now consider this as with the help of this yellow color, I'm going to draw a structure. So this structure, consider this as the axonal nerve ending, that is the terminal button, it is also known as terminal button, where the neuron terminates. Okay, now, and this one, with the help of this brown color, I am going to draw a muscle cell. Okay, a muscle cell where it is uniting with this neuron, a muscle cell. Okay, a just rough diagram of this one. This is a muscle cell, okay, where it is uniting with this neuronal cell membrane. Okay, so this is the muscle cell, and this is the neuron or the axonal end, or this is the nerve tissue or neuron. more specifically the axon or terminal button. It is also known as terminal button. So as I have said you in the introduction itself, this neuronal ending or the axon has plenty of synaptic vesicles. Okay, synaptic vesicles. So nearly 3 lakh such vesicles will be present in each neuronal terminal ending. Okay, so these are termed as synaptic vesicles. Now these are protein in nature, so they are generally produced, okay, or manufactured, where it is produced here in this cell body. So this is the axon here. Okay, what is a nerve? Nerve is nothing but the bundle of axons in the peripheral nervous system. If the bundle of axons are present in the peripheral nervous system, we simply call it as a nerve, or the bundle of axons, if it is present in the CNS, that is the brain or the spinal cord, then it is called as tracts. We have ascending tracts or descending tracts. So now in this neuronal cell bodies where there is a genetic material, that is the nucleus, DNA will undergo transcription and form the mRNA, and mRNA is translated to form the proteins. These proteins are formed, synaptic vesicle proteins are formed here, and through this exoplasm it is transmitted to the neuronal terminal ending. So nearly 3 lakhs of synaptic vesicles will be present at the end of the neuronal ending. Now in this vesicle there are acetylcholine molecule. Okay, acetylcholine molecule will be present, and each of the synaptic vesicle will be containing around 10,000 such acetylcholine molecules, which is a very important neurotransmitter. Now this acetylcholine molecule, how it is formed? So this acetylcholine molecule, which is very important structure for the neuromuscular transmission. So formation of the acetylcholine, it is very important to know. Basically this acetylcholine is formed by 2 structures known as acetate or simply acetyl-CoA, and also by one chemical known as choline. So acetyl-CoA and choline will unite to form this structure acetylcholine. So what is the source of this acetyl-CoA? So you know in the neuronal terminal endings there is mitochondria, which is a source of energy which forms ATP. So through this mitochondria this acetyl-CoA is also synthesized, or also known as acetate, okay, through the mitochondria. And choline, it is a nutritious substance. It is derived from the GI tract, that is the food whatever we eat. Through our GI tract, this food is ultimately transmits or produce the choline, which goes into the circulation, and from circulation through choline receptors, this choline will enter into the neuronal cell. Okay, neuronal cell. So this source of choline and acetyl-CoA we have discussed, and with the help of very important enzyme known as choline acetyltransferase, one enzyme which will catalyze this reaction, choline acetyltransferase, which will catalyze this reaction and combine this acetyl-CoA to choline. This forms a very important neurotransmitter known as acetylcholine. Once this acetylcholine is formed, it is rapidly transmitted into the synaptic vesicles, and as I already said, each synaptic vesicle will be having around 10,000 of this acetylcholine molecules. Okay, now this is the general structure and formation of the acetylcholine. Now this stored acetylcholine will enter the synaptic clefts. What is synaptic cleft? The space between this presynaptic and the postsynaptic membrane. Presynaptic membrane means axonal membrane, postsynaptic membrane means muscle membrane. Okay, so this is generally around 20 to 30 nanometer wide, known as synaptic cleft. Okay, synaptic cleft. So in this synaptic cleft, how exactly this acetylcholine molecule will get transmitted, and this chemical will causes the depolarization of the muscle. So initially what will happen, the action potential of the nerve, okay, when the neuron is depolarized, okay, those who don't know what is the action potential of the nerve, I request them to go back to my previous video of action potential of nerve, or just simply go to the playlist of neuromuscular physiology. You can find that lecture video of action potential. So the action potential, basically the depolarization and repolarization. So depolarization, current passes to this terminal ending, and this depolarization current, when it passes, that is generally the nerve will have a resting membrane potential of minus 70 millivolt. Okay, so it will reaches to around plus 35 millivolt. So when it reaches to around plus 30 millivolt, on this surface, on this membrane of the neuron at the terminal ending, there are very specialized channels known as voltage sensitive or voltage gated calcium channels. Voltage sensitive or voltage gated calcium channels are present. Okay, so whenever the depolarization wave, that is the interior of this cell, becomes positive, that is the cell becomes excited, the action potential signal is passed through this terminal knob or the synaptic knob or the terminal end of the axon, then this voltage gated calcium channels opens, and through this voltage gated calcium channels, calcium from the ECF, that is the extracellular fluid, will now enter inside this cell. Once the calcium enters inside this cell, now what this calcium will do? The calcium will now drag the synaptic vesicles towards this towards this cell membrane, okay, near the neuromuscular junction, towards the cell membrane, and ultimately it will drag it outside, and the neurotransmitters which are present, that is acetylcholine, which is formed and stored in the synaptic vesicles, are ultimately released out into the synaptic cleft space that of the neuromuscular junction by the process of exocytosis. Okay, you know what is endocytosis, what is exocytosis. So it will by the process of exocytosis, this acetylcholine is released. Now how this calcium will causes or drag the seminal vesicles into the towards the cell membrane, and it causes the exocytosis. If you want to know the details, let me show you here. So how exactly it will happen. So again, consider this as a neuronal cell membrane, and here in this neuronal cell membrane there are synaptic vesicles, right? Now on this synaptic vesicles, which is a protein in structure, there are certain proteins which are known as snare proteins. Okay, I won't go in detail about the snares. There are two types of these snare proteins. One is V snare proteins, another one is T snare proteins. V snare means the proteins which are present on the vesicle, that's why V snare. Okay, so there are certain proteins which are present on this vesicle. So let me name them. There are many, I'll just name the very important one. Synaptobrevin, V snare proteins. Okay, synaptobrevin, or one more, synaptotagnin. Okay, synaptobrevin and synaptotagnin. Okay, synaptobrevin and synaptotagnin, these are the V snare proteins. Now also there are T snare proteins which are present on the membrane, which are attached to this or present on the membrane of this neuronal ending. These proteins are like SNAP25 or syntaxin. Okay, syntaxin. So generally they are not attached to each other. Okay, they are separated, but once the calcium ions concentration increases inside this ICF or this neuronal ending, this calcium causes the these two snare proteins to unite, and ultimately when they are united, these synaptic vesicles are dragged and get attached to this membrane, and ultimately by the process of exocytosis, the acetylcholine molecules are released into the neuromuscular junction, that is the synaptic cleft, that is the space. Okay, now this, whenever this so many synaptic vesicles together, they will be they will causes this exocytosis and release of this membrane. So on this muscle cell, there is one of the very important structure known as ligand gated ion channels, or ligand sensitive ion channel, which is known as acetylcholine receptor. This is known as acetylcholine receptor, which is a ligand gated receptor. More specifically, it is the nicotinic acetylcholine receptors, which is a pentameric protein. Pentameric protein means this are made up of 5 proteins, okay, 5 protein subunits: alpha 1, alpha 2, beta, then delta and gamma, which can be simply written as alpha 2 and beta, delta and gamma. That is, these 5 proteins will form a tubular structure. This is known as nicotinic. Why it is called as nicotinic acetylcholine receptor? Because this receptor can be stimulated by one drug or chemical known as nicotine. Okay, so on the surface of this there are active sites on which acetylcholine can bind. So whenever this acetylcholine binds on the surface of this receptor site, then it was generally closed earlier, it will get opened. Okay, also nicotine can also stimulate this receptor and it can open it. Okay, that's why it is known as nicotinic. There is one more known as muscarinic. We'll discuss about that in some other time. So now these nicotinic acetylcholine receptors which are present here, when this acetylcholine binds on the receptor sites, so what will happen is this acetylcholine bind, that's why it is called as ligand gated. Ligand means chemical gated. It is not voltage sensitive, it is sensitive to do chemical change. So whenever it binds, it will open, and the cations, mainly the calcium from extracellular fluid, not the calcium, sodium. Okay, even the calcium will enter, but mainly the sodium will enter here. Okay, sodium will enter through this channels. Okay, so through the acetylcholine receptors, mainly the cations like sodium will enter from the ECF, that is the synaptic cleft, to the this one muscle cell. Now if you see the electrical activity of the muscle cell, if we draw a diagram, on the x-axis that is time in millisecond and on the y-axis voltage in millivolt. Okay, so you know that at the resting condition it is around minus 90 millivolt. That is, the electric current will be minus 90 millivolt. That is, the interior of the muscle cell is negative at the resting condition. Okay, minus 90 millivolt. Now what happens is, when this cations like sodium enters here, on some even the calcium when enters here, what happens? It increases the positivity. And it causes some membrane potential difference. Okay, some membrane potential difference, but it is less than the threshold potential. You know, the threshold potential is around minus 55 millivolt in the muscle approximately. So some membrane changes occur and the positivity causes the membrane potential to reach to minus 80, minus 75, but it is less than the threshold potential. So it will not trigger the action potential, and ultimately this potential will die. So these electrical changes after the opening of the ligand gated acetylcholine receptors and entry of the cations, what it will causes, it causes some electrical activity. That electrical causes the local potential. So this is a local potential, and the name given to this local potential is end plate potential. Okay, EPP, or end plate potential. Okay, which is not the action potential. It is a non-propagative in nature. It does not obey all or none law. Okay, the difference between the graded or local potential and action potential we'll discuss in some other video. It is one of the example of a local potential. So what is this potential? So whenever this acetylcholine receptor opens and cation enters here, it causes certain voltage fluctuation in the membrane of the muscle cell, and that muscle's voltage, it is known as end plate potential, which is not an action potential, and it dies out. When this end plate potential reaches the threshold potential, then only the action potential will take place and the muscle gets depolarized. Okay, now before that there is one more term known as miniature end plate potential. It is a small local potential which is caused by the opening of the single vesicle. Okay, whenever there will be multiple vesicles will be opening, and nearly 125 such vesicles, 125 vesicles are opened, and it is transmitted to the synaptic cleft and acetylcholines are released. If suppose only from one vesicle one unit of acetylcholine molecules are released, and they bind to these acetylcholine receptors and causes a voltage difference, and that voltage difference is very less, just around 0.4 millivolt, less than 1 millivolt. Okay, it's a small fluctuation. That small fluctuation, which is the minimal current, okay, occurs, that is called as miniature end plate potential. Below that there is no electrical current, that is miniature end plate potential, and this miniature end plate potential summed up to form the end plate potential. And finally this end plate potential can reach up to the threshold potential and causes the depolarization. So when this end plate potential reaches the threshold potential of minus 55, that is from minus 90 to minus 55 millivolt, then there are voltage sensitive sodium channels which are present on this muscle membrane. Okay, so this voltage sensitive sodium channels opens and the interior will become positive, and the sodium ions enter here, which causes this depolarization, and finally potassium channels are also open, which causes the repolarization. We'll discuss about it in the molecular basis of muscle contraction, how exactly this depolarization and repolarization takes place and the muscle will contract. Right now in this particular topic of neuromuscular junction, we have discussed about how exactly the action potential which reaches the neuronal cell ending is transmitted into action potential of the muscle cell. Then what is the fate of this acetylcholine? So this acetylcholine which was released, this acetylcholine can be broken down by very important enzyme, acetylcholine esterase. Okay, acetylcholine esterase. So this acetylcholine esterase will break the acetylcholine into acetyl-CoA or acetate and choline. So choline can be again retaken back by this cell, okay, through the choline transmitters. Choline can be again retaken back into this cell, and acetate is absorbed back, and some amount of the acetylcholine will be diffused out of the synaptic cleft into the ECF. Okay, so this is the fate of the acetylcholine. Now why we need to know all this molecular basis? So it is very important because in diseases like myastheniagravis, there are autoantibodies, that is our immune system will produce certain autoantibodies against this acetylcholine receptors, more specifically nicotinic acetylcholine receptors, and more specifically type 1, and destroy this. So when it gets destroyed, even though acetylcholine released here, it will not causes the depolarization of the muscle membrane, and the muscle will not contract, okay, which leads to the muscle weakness and ultimately paralysis. That is the myastheniagravis. Similarly, in one disease known as Lambert-Eaton syndrome, there are autoantibodies against this calcium channels in the presynaptic membrane. This is known as presynaptic membrane, that is the nerve membrane. This is known as postsynaptic membrane or the motor end plate, muscle membrane. Okay, now these calcium channels on the presynaptic membrane, when they are destroyed by the autoantibody, they are, this disease condition known as Lambert-Eaton syndrome. Again, there are certain drugs and toxins like botulinum, tetanus, and some drugs, neostigmine and all, which will act on these enzymes and these various receptors which are present in the neuromuscular junction, which we'll discuss in the some other lecture video. So right now, just to summarize the today's topic of the neuromuscular transmission, how exactly the neuromuscular transmission, that is the electrical event, will causes the transmission of this signal from the nerve to the muscle. So when the action potential reaches the neuronal terminal ending, voltage gated calcium channels open. This calcium channels causes the synaptic vesicles to drag and come near the synaptic membrane, that is and it, due to the presence of exocytosis, acetylcholine is released, and this acetylcholine causes the opening of this voltage ligand sensitive or ligand gated cationic channels or receptors are known as acetylcholine receptors, and this sodium causes end plate potential, and ultimately when it end plate potential reaches the threshold, opening the voltage gated sodium channels, and action potential is generated in the muscle cell. So ultimately electrical energy gets converted into chemical energy gets converted into electrical energy. This is how a signal from the nerve is transmitted to the muscle. Hope you like our video. Please subscribe our channel Doctor's Corner. Thank you.